good afternoon or almost evening, depending where you are already. Thank you for inviting me to present today our work on developing antisense oligonucleotide-based therapies for Stargardt disease that we are conducting here in Nijmegen in the Netherlands at the Radboud University Medical Center. Before I start, some disclosures. So several of the co-authors in this abstract and in this work are inventors in patents describing the use of antisense oligonucleotides to treat inherited retinal diseases. And I will start first with the eye. So the eye is the organ that allows us to see what's happening around us. And this happens because at the back part of our eye, we have a complex neuronal tissue called the retina that contains several layers of neurons. And one of the most important type of neuron are the photosensitive cells, the rods and the cones, that are in charge of capturing this light, converted into electrical and chemical signals that travel to the brain, where the information will be created while processed, and then an image will be created. So in patients that do not have or that have a defect in this photoreceptor or photosensitive cells, what happens is that they gradually lose the vision until they become completely blind. And when this photoreceptor cell death is caused by a genetic cause, we have what we call inherited retinal diseases, or IRDs. And these IRDs are rare monogenic diseases, overall affecting 1 every 3,000 individuals worldwide. And they present a high genetic heterogeneity in the sense that we know nowadays mutations in more than 250 genes that cause any type of IRD. And here in this graph, what you see is the different subtypes of IRDs in each circle and the genes that have been associated with those diseases. And as you can see, there's a lot of overlapping. So overall, that also means that each mutation is in a low recurrence, and each gene contributes in a small amount to the total 1 in 3,000 cases. So this is something to keep in mind. And despite all the knowledge that we already know and we have gathered during the last years, around 35 to 30% of our patients still do not have any genetic diagnosis, meaning that they are not eligible for any type of personalized treatment that could be developed because the cause of their disease is not known. But IRDs, in the positive side, what they have is that they are progressive loss of vision. So they are progressive diseases. So we have a window of opportunity. Usually the first symptoms appear during the first or second decade of life, and usually at the age of 40, 50, the patient can become completely blind. So we have around 20 years where we can act. So what it has been done already for antisense oligonucleotides for IRDs. So far, 7 different genes have been targeted. Some of them are in different clinical trials or in preclinical stages. And what is interesting is that 5 of them are modulating splicing with ASOs, but the other 2 use more the transcript degradation or gap mods in order to degrade a mutant transcript. And today I will be focusing on these splicing modulation possibilities that ASOs offer. So when we think about splicing defects, it's good to start mentioning, okay, if we have a gene of 3 exons, like the one that is depicted in this slide, in the normal situation, the introns will be removed and the exons will be pasted together, and we will have a protein that will be synthesized from this mRNA. But we can have a mutation in the splice site, and this can cause, for instance, exon skipping, so the exon 2 will not be included. And this will disrupt the reading frame and eventually create a protein that is not functional or degrade the transcript via nonsense mediated decay. But we can also have exon elongation. So a novel splice site in the intron will be used, and this exon 2 will be longer. And that also means that the reading frame will be disrupted. Again, the protein will be not functional. And we have the most extreme case, that is the intron retention, where the entire intron is retained and no splicing occurs. And again, it happens the same that I mentioned before. But there are other types of mutations that during many years we have ignored, mainly because we thought that all the possible disease-causing mutations were in the exons or the splice sites that are the deep intronic variants. And these deep intronic variants, as the name already indicate, are in the introns, usually deep in the intron, and what they usually create is an over splice site that will lead to the insertion of a pseudoexon, so a piece of exon that will be recognized as a piece of intron that will be recognized as an exon, and that means that the reading frame will be again disrupted. And in most of the cases, these pseudoexons already contain a premature stop codon in their own reading frame. So nowadays we have the technology to check this, but during many years, of course, we were checking only the exons using whole exome sequencing. Nowadays, we use whole genome sequencing or targeted approaches where we sequence the entire gene from the beginning till the end. But this sequencing is challenging because introns also have very repetitive motifs, and sometimes they are not well covered, for instance, in whole genome sequencing. And that means that it also poses a challenge to find these mutations. But once we find them, the next challenge that we have is how to interpret them, because there's little information on how these introns behave, what they can be or not. But nowadays we have also AI softwares like Splice AI, where we can indicate, okay, we have this new variant, and the program calculates whether there's the possibility of a pseudoexon inclusion by checking whether, first of all, a splice site is created, but also whether in the neighborhood, let's say 200, 300 base pairs away from this variant, there could be another acceptor or donor site that could complement this new site that has been created, because at the end, we need to have an acceptor and a donor close by to create a pseudoexon. So that helps a little bit, but in IRDs, we still have an extra challenge, and it's how we are going to validate these in silico predictions. So most of the genes that I've shown you in the previous slide are only expressed in the retina, and we cannot go to the patient and ask them, oh, can we get a piece of your retina? We also cannot culture. The retinal cells are neurons, so they do not divide. So we need to also look for other models. And once we have these models, we also can then try to correct this splicing defect with an antisense oligonucleotide, for instance, but we also depend on the cell type. So once to circumvent this, we use two different models. The first one is what we call the artificial model, that is using a midi-gene, and a midi-gene is just the extended version of a mini-gene. In a mini-gene, usually you have a small piece of genomic regions flanked by two known exons. And in this case, in the midi-gene, we realized that the more context we put, the more reliable the results are. So basically, we clone a significant big or large piece of genome, so usually around 8 to 10 kb. And we have also the flanking regions. And then with that, we can introduce the mutation that we want by site-directed mutagenesis and transfected in cells like HEX cells that are transfected really well. The other source is to use fibroblasts from patients or blood cells that we can reprogram to induce pluripotent stem cells. And then after while using several protocols, we can develop retinal organoids or photoreceptor precursor cells that allow us to increase the expression of those genes that are only expressed in the retina. So today I'm going to focus on ABCA4 and Stargardt disease. And Stargardt disease is one of the most frequent IRDs. In particular, it's a juvenile macular dystrophy affecting 1 in 10,000 individuals. It's like all the IRDs, a progressive disease, and it's caused by biallelic mutations in ABCA4. And that means that it's an autosomal recessive disease and that both alleles need to be mutated. And if we look what ABCA4 is, it's a transmembrane protein that acts as a transporter. It's one of the larger genes that we cause IRD. It has more or less a cDNA of 7 kb with 50 exons, and so far more than 2,000 unique variants have been reported. As the function, well, it's a transporter. In particular, it's an ATP binding cassette to family A member 4, and it's crucial for the visual cycle, because it's part of the, it transports the retinal from the lumen to the outside of the lumen where the opsins can take it. So if we look at the genetic part, what my colleagues in Nijmegen, the group of transcribers, have done is sequence more than 1,000 patients. And overall, what we have seen is that half of those genetic pathogenic variants are missense variants. But then there's a significant proportion, 21%, so 1/5 of all the variants that affect splicing. And in particular, 11% of all these variants are deep intronic variants, so they create a pseudoexon. And this is what is depicted in the plot, in the graph. So here we have all the variants that we have discovered, and we can see that some of them are highly prevalent compared to others that maybe there's only 1 or 2 cases. And what we have done is target all these mutations. So far we have developed antisense oligonucleotides for all of them. So if we look where they are located, the deep intronic variant seems to be more frequently in those introns that are rather large compared to others, but there's no significant area where we can say there are more the intronic variants, the deep intronic variants than others. But then we also have what we call near exon variants. And these near exon variants are variants that are close to the exon, and usually they do not cause a pseudoexon insertion, they cause a exon elongation. And what we have done is use first this mitigene system for validating those variants, and I will only give one example instead of going through all of them. So again, we have this mitigene flanked by the 2 exons of the rhodopsin and a big piece of the genomic region. We insert the mutation, we transfect it in HEC293 T cells, and then we do RNA analysis. And what we can see for this particular variant is that in the wild type situation, we only have 1 transcript that corresponds to what we would expect. And then in the one with the variant, there are 2 bands, and the higher one and most intense contains a pseudoexon of 56 nucleotides. So using this system, we have been able to analyze all these variants and also test oligonucleotides. And this is a work that has been done by 2 of our PhDs, Nuria and Tom. So here an example for a near exon variant. So what we try to do is splicing redirection. We have a variant that introduces a 36 nucleotide exon elongation. We designed the oligonucleotides in a way to block the new splice site and allow the splicing machinery to go back to the original splice site. And in that sense, while here we have the result, we can see that in the wild type, nothing is happening. It's for these 4 lanes, but then when we go to the mutant, we have the insertion. You can see that the band is bigger than the control. And when we deliver the ASOs, in both cases, we can see that the splicing is redirected. In the case of the second ASO, we can see that there's still a little bit of incorrect transcript present. And then we also use a scramble oligo, where again, we don't see any effect. And this graph is just a quantification of these gels that we did. So for these near exon variants, we have been able to correct all the variants that had the new, created a new splice site at the donor site, let's say. But for those at the acceptor site, so at the 5' of the exon, those were a little bit more challenging. And in particular, the only variant that we have not been able to target was one variant that was at the 5', creating a new acceptor site. And we think that the reason why it did not work is because that variant was also disrupting the branch point that is needed for the splicing. So even if we targeted that splice site, the splicing could not be conducted because the branch point was not there. But then we have the easiest approach, that is the deep intronic variant. And in this case, we have a 127 nucleotide pseudoexon. And we have designed 2 oligonucleotides, in this case, targeting the strongest splicing enhancers, as it was described by Annemieke in the guidelines that she published several years ago. And we were able to, again, test that. So here we have the wild type. We see that there's no band in the wild type. And then when we go to the mutant, we see clearly two bands. When we sequenced them, there were two independent pseudoexons. One was shorter than the other one. But after the treatment with ASO, the band disappears, and we can clearly see that the lower band increases in intensity. So we have been able to redirect splicing. And with the scramble oligo, no difference have been observed. And again, here the quantification where it's clearly visible that the pseudoexon has disappeared. So we have done this for the 25 variants that I mentioned, and now what we are doing is try to translate this to the clinic. In the case of the N of 1 cases, we are doing it together with the Dutch Center for RNA Therapeutics that was established 2 years ago to bring all these N of 1 cases to the clinic. And we are looking which AON is more promising for that. And then for the most recurrent ones, we are trying to do it in the conventional way. But then this mini gene system not always is reliable, and we need other sources of cells or material to test. And in this case, we were studying these 4 mutations in the intron 36. And basically, this one that I highlight, the M9, when we did the mini gene approach, we could see that, well, compared to the other ones, the amount of pseudoexon was quite low. So you can see that M4 and M6 is really clear, M11 also really clear, but here there's a shadow that if we quantify, it's only 10%, which for a recessive disorder is quite low. So, but this mutation is highly recurrent, so we were sure that it should do something. So we were able to obtain cells from the patient. We reprogrammed them to iPS cells, and we differentiated them to photoreceptor precursor cells. And indeed, what we could do is that, what we could see is that this mutation actually creates a pseudoexon, and it's really clear. So let me guide you through it. So we have the control cells, and we treat them with cycloxamide to block the nonsense-mediated decay. And we can see that in the wild type, in the normal situation, there's no band, but if we block the nonsense-mediated decay, there's a little bit of band. So that happens because there can be some pseudoexon insertion because the cryptic splice site, it was already there. But then we can see that in the M9, already without cycloxamide, so without blocking the nonsense-mediated decay, we already see this pseudoexon. And when we block the nonsense-mediated decay, the band even increases to a 1-to-1 ratio. And it's important to mention that this patient is compound heterozygous, meaning that only one of the 2 alleles carry this mutation. The other is a missense mutation in another allele. Therefore, that means that there's the effect of this mutation is highly penetrant based on the intensity of the bands. So knowing this, we decided to use these PPCs to test our oligonucleotides. And these PPCs basically is just going from iPS cells and then differentiate them for 30 days. And if we're going to go to retinal organoids, we need to do it for 150 days. In that sense, ABCA4 is already highly expressed. We designed 4 oligonucleotides targeting different regions of the pseudoexon. And here is what we see when we do the experiment. So we can see that in the wild type, there's a little bit of pseudoexon, but nothing compared to what is in the mutant. And we can see that AON6 and AON7 actually increase the lower band and decrease the upper band, indicating that the splicing has been redirected. So far, I have shown you results in the midi-gene system or artificial system using human-based models, which are more reliable for our experiments. But what's next? So how can we assess the ASO efficacy and biodistribution in the retina? And that was work that our postdoc, Irene Vázquez Domínguez, was doing. So what we have done is investigate the uptake in different cell types For that, we selected 4 different cell types: rods, cones, bipolar cells, and RP. All of them can be involved in disease. And we selected markers that are only expressed in those genes and then target them with different chemistries of oligonucleotides, so 2-MOE, 2-MOE, and PMO. And what we saw is that so far the 2-MOE, as also has been described in literature, in vitro, has been the most successful one in redirecting splicing. And here just an example how we did it. We just took the entire mouse gene, we clone it, and we deliver to the cells. And then we deliver, in this case, it's AON2 was the most promising one. And we can see here that the upper band, that is the wild type, has been decreased and exon 4 has been skipped. That is what we were looking at. So we wanted to modify splicing to quantify which chemistry was better. So then we move in vivo. And in vivo, what we, and this is the example for GM6, which is a bipolar gene, cell gene. And then here we did the same. We injected 5 replicates, and this is just a representative gel and the average of the 5 replicates. So what we did is inject the left eye of the mice with PBS and the right eye with the oligonucleotide in each of the 3 chemistries. And we can see again that the 2-MOE and the 2-MOE phosphorothioate were the most efficacious one, while the PMO was not. And in fact, the PMO also caused some cataracts and toxicity in the eye that we are now investigating whether the retina was also affected or it was only the lens. And here we have the scrambled oligo where we see that no splicing occurs. So basically we can for sure say that it was caused by the sequence itself, not by the chemistry. So overall, to conclude, deep intronic variants explain most of the unsolved cases in our Stargardt cohort. Pseudoexon insertion is the main splicing defect identified, and splicing, splice assays in an artificial system are very useful to test, to validate whether a splicing defect is present or not, but also to test ASOs. However, several deep intronic variants show a retina-specific splicing defect, and for that, it's better to have patient-derived cellular models because they have the entire context of the gene. So in our case, in the MIDI gene, it's always a part of the entire gene, and we don't know how the RNA structure can play a role. So in total, we have design and test more than 25 ASOs for different intronic variants. And based on our studies targeting 8 different targets, seems that the 2-MOE phosphorothioate is both the most effective chemistry, both in vitro and in vivo. So with that, I would like to acknowledge the entire Colin and Garanto lab for their work, also the funding bodies, all the collaborators that have been involved in the ABC for Stargardt disease story. And of course, all of you for listening in. And if you have any questions, I will be happy to answer them.